but it's not enough at the end. And I just want to give you one example of the concept of physiologic reserve. And I already told you your kidneys have a huge reserve to reclaim far more glucose per minute than what a healthy person has. Seems to me I mentioned another thing, physiologic reserve, but here's an example of physiologic reserve. Cardiac output is how much blood is ejected from your heart every minute. So that's your heart rate times your stroke volume. Heart rate, how many contractions per minute. Stroke volume is how much blood is being ejected with each contraction. Those two things multiplied together gives you cardiac output, the amount of blood that leaves the heart every minute. So, you know, you're sitting here, and I put these numbers kind of high. Let's say your heart rate is about 70 beats per minute, and with each contraction, with each beat, your heart's ejecting about 70 mils of blood per minute. Okay, that's pretty typical, and has a cardiac output right around 5 liters per minute. That's very, very typical. But you know when you begin to exercise, your heart rate goes up. What you can't feel, but it's happening, is your stroke volume. The amount of blood that's ejected with each beat goes up as well. Heart rate in people can go up 2 to 3-fold higher. Stroke volume can double in many people. And this is with healthy people. This would be a healthy young person. This would be a healthy young person. So you can see that it's going from about 5 to very close to 30, close to a 6-fold physiological reserve. You have this much to play with when your body is challenged, and you need a higher cardiac output to put more blood flow to the tissues so they can take up more oxygen and do more work. So there's a huge physiologic reserve. With age, this reserve gets less and less. With, let's say, a myocardial infarction, and you lose some of, let's say, your left ventricle, your stroke volume is going to go down. This is going to become less and less. So there will be people that you treat who basically them just getting up to walk to the bathroom is about maximum physical activity for them. In many of those cases, they've just pretty much used up all their physiologic reserve. And there are some patients you're going to have that they're just going to be, you know, will be bound to the bed because they don't have much physiologic reserve. But I think you can see that if one loses this reserve and then something happens and a person's body is challenged and they need a higher cardiac output, but they no longer have it, there's going to be symptoms. There's going to be symptoms that the patients report. Signs you'll definitely see.
Does that just go hand in hand then with being fatigued easier?
Pardon me?
Does that just go like hand in hand with being fatigued easier? Like you said, if you get up and go to walk to the restroom, it's harder. Is that because you're not getting enough oxygen?
Yeah, exactly. And well, believe it or not, you run into CO2 problems more quicker than oxygen problems often because, you know, we think we breathe and our heart beats and injects blood to supply tissues with oxygen, but as important, actually even more important, is to get rid of CO2 because CO2, the retention of CO2 very quickly within seconds makes you acidotic. And when you become acidotic, guess what? A lot of those proteins in your bodies start to dysfunction. So we breathe, we have aerobic metabolism for a lot of things to make ATP. You know, we breathe to get an oxygen. How do we use oxygen? That final step of the electron transport process when we're making ATP by aerobic metabolism. We need oxygen there. So if you don't have enough oxygen, you're not going to be making enough ATP. But as importantly or more, you're not getting rid of CO2. And we're going to see CO2 accumulation within seconds to minutes makes you acidotic. Okay, so anything else? Anything else about homeostasis? Yes?
I had a question about the pepsinogen.
Oh, yeah.
Which cells did you say were these?
Chief cells, gastric chief cells. yeah, you'll get that, and yes, I'm happy to fill that for you. So, we're going to talk about membrane dynamics. Some of this we'll go through kind of fast, because you've heard about membranes since you were in probably 3rd or 4th grade, but there are some physiochemical properties of membranes that really regulate some functions, and our body has to figure out ways of going around those properties to get molecules in and out of cells, okay? So, we're going to look at very quickly the structure of membranes, and then we'll look at the functioning. I was hoping to, we might not quite get to membrane transport. Okay, so, the first concept I really, really want you to appreciate is this concept of gradients. A gradient just means a difference between two compartments. Some days this room will be warm, the hallway will be cool. That's a temperature gradient. Just means there's a difference between two compartments. Well, our body makes gradients, a difference between two compartments, in a lot of different ways throughout the body. And the body spends a lot of energy to set up these gradients. And why? You can unleash. Think of the gradient as almost stored energy. It is stored energy. It's not ATP, but it is a form of energy that once you release it, you can do work. You can drive physiological processes. Now, all of you have had some physiology, so you know that, for example, let's say this is your left ventricle, this is your aorta, so here's your aortic valve. When that left ventricle contracts, it pressurizes the blood, and during diastole, the blood pressure in your aorta drops and drops and drops, and fluids flow from an area of high pressure to an area of low pressure. So by generating that high pressure, blood is going to flow out of that heart and all through the vascular tree. So a pressure gradient drives blood flow. Oh, okay. Those of you who had some respiratory physiology, you know that we breathe by something called negative pressure ventilation. I'll let you finish writing so you can look at my amazing artistic representation here. So when you make an inspiratory effort, your diaphragm dives down. This actually makes the pressure in this intrapleural space negative. That causes your alveolar walls to distend outward. What does that do? It causes your alveolar pressure to become sub-atmospheric. For ventilatory mechanics, we call pressure in the atmosphere to be zero. Guess what? We have a pressure gradient. Here out in the atmosphere, zero, that's higher than this -2. Air is a fluid. Fluids flow from high pressure to low pressure. We have air coming in. And then all we have to do is relax our diaphragm. It's going to snap back, and what's that going to do? It's going to pressurize that gas, and air flows out passively. We don't even have to contract any muscles during normal quiet tidal expiration. We just relax. And because of elastic recoil, it drives that air out. So pressure gradients cause air and blood to flow. Okay? Concentration gradients, some cells use 90% of the ATP that they produce for creating a gradient between one area and another area. So think of these as like ions. You know, those could be sodium ions. Could be. And that's a concentration gradient. There is a large concentration of one ion here, much lower concentration there. We're going to see that concentration gradients provide us the ability to do a lot of things, including transporting other molecules across that membrane. So, just to let you know that a couple of years ago, I was going to present something on this in January, the end of January, and the day before, man, I'm telling you, there was a high pressure system up in eastern Montana and down around Arkansas. It was low pressure, and we had 40-mile-an-hour winds all day long. So that's a pressure gradient, and why I didn't have that before the concentration gradient, I don't know. I put it in the wrong place. But this is an example of pressure gradients, whether it's in your bloodstream or in your lungs, it causes fluids to flow. I put it in the wrong place. So, back to concentration gradients. Here is a relationship, and your book uses slightly different numbers, and interestingly, your book uses slightly different numbers in different areas of your book. I have people remember these concentrations for a reason. They're physiological, and like your book in a lot of places puts this at 145. But these are easy to remember, and you'll do okay with them. For sodium, in the extracellular fluid, I'm going to tell you, just think about, you have about 150 millimolar of sodium in the extracellular fluid compartment. Inside of cells, it's much lower. It's 10-fold lower. It's 15 millimolar. Now, you're going to see clinically that the normal range for glucose in different places have it different. It might be 135 to 148. But I'm going to ask you for right now, stay with this 150 because that 10-fold difference is going to make your life a little easier later on. So there's a lot of sodium outside the cell. With potassium, there's a bunch of potassium inside of the cell compared to outside of the cell. About 140 millimolar inside of a cell than outside the cell. So you can see two different chemical concentration gradients there, don't you? And both of those can do work for us. They can drive physiologic processes. You can open up sodium channels or have sodium latch onto a protein, and when it goes from high to low, it'll do work for us. Same thing with potassium. Now, a lot of students over the years have told me, boy, I just keep getting sodium and potassium flipped around. Where is one high? Where is one low? Where is one high? Where is one low? 4 years ago, one of my first year dance students said, Dr. Crouch, she was almost embarrassed to tell me how she remembered it. And I just have my students, hey, if you have some quirky way of remembering something, tell me. And I put it in the notes from that point on with their picture, and they become, in physiology class, they become physiologically immortal, right? And so, here's a little story about cake. Cake is probably, I remember it, Dr. Crouch, it's the ocean banana. What on earth is the ocean banana? Dr. Crouch, think of a beautiful ocean view. Okay, Caitlin, I'm with you. And then put a banana there. Oh! What's in the ocean? Salt, sodium. What's inside a banana? We all know it's potassium. You remember that picture? You'll always know. Sodium's highest on the outside, potassium's highest on the inside. And I have a dozen of these quirky things for biochemistry and physiology that students tell me every year. Wow, something popped up on the national board exam, and I had it down cold because of something that a student told me, and I have stuff that I have in my notes from students 20 years ago. And anytime you come up with something, you let me know because, you get your face on the slides. Not just here, but over the College of Dentistry too. Okay, so do remember this now and forever. Okay, so there's another type of gradient. These three, there's a lot of different gradients. pH gradients, temperature gradients, calcium gradients, but, well, yeah, that go under concentration gradients. But there's another one called electrical gradients. Normal living cells, compared to the extracellular fluid by convention is considered 0 millivolts. The interior of living vital cells, it's electronegative. And these are millivolts, so don't think this is a big difference in electrical charge, it's not. But some cells at rest are -10, some are -30, some are -75, some are like -90, okay? Depends on cell type and cell size. But at rest, you have this electrical gradient. This electrical gradient is so important for graded potentials, action potentials, helps with synapses, helps with release of hormones from cells. It's amazing. Just a little electrical blip will tell a cell what to do and when to do it. Okay? So, let's look at membrane organization. And we'll see if we can get a little bit into transport. So here is your slide 3.6. You've seen a slide like this so many times in your life. After we go through this, I really want you to look at it and appreciate some things about it. You have phospholipids. What on earth is a phospholipid? Here's a glycerol backbone right here. And here is a fatty acid with its hydrocarbon tail. Here's a fatty acid with its hydrocarbon tail. And what is on the head of these phospholipids? Well, it's a charged substrate here. So we can see a couple of charges there, a charge there, a charge there. So these are interesting molecules because these fatty acid tails, they are hydrophobic. They want to get away from water. Charged heads are hydrophilic. They want to hang out with water. And you have both of those things in one molecule. So this part, the phosphate heads, they want to hang out with water. That's what you have here in the extracellular fluid. These heads want to hang out with water. That's what you have in the intracellular fluid. These tails, these fatty acid tails, and they interdigitate just a little bit more than what they show here. They are a barrier. They are a barrier. And they just want to hang out with fat. And you see some cholesterol in here. And they really don't let charged particles through because this is a permeability barrier. It will let electrically neutral things through, but things that are polar means charge. I'll show you a whole bunch of things to come. So here we have some integral proteins. Integral proteins are proteins that are embedded within the membrane, and some are just in part of the membrane. Some actually completely cross the membrane. That would be called transmembrane protein. Trans means across. And then there are important proteins that line the inner leaflet. Those are peripheral proteins. But so these are just chains of amino acids, and you can bet that the amino acids are in this area of this protein have functional groups that like hanging out with fats. The amino acids up here have functional groups that like hanging out with water. That's why these proteins fit the way they do. So once we go through all the things we're going to talk about, think about what this means with why is this polar, why is this non-polar? And that's what is supposed to mean here. So I draw things, simple line diagrams because people tend to remember simple things more readily. So what this really is with these fatty acid tails coming together, they are a hydrophobic permeability barrier. It doesn't allow charged molecules to go through very easily. So the phosphate heads, they are charged, so they like hanging out with water. Water happens to be a charged molecule. I'll show you a picture on that. But these tails are anything but. They are hydrophobic. These heads are hydrophilic. And when you have hydrophilic and you can say that's amphipathic. I don't know if you guys need to know that. But the fatty acid tails are electrically neutral, meaning they're uncharged, hydrophobic, and lipophilic. They're creating a barrier against hydrophilic, lipophobic charged molecules. So this is a barrier. Fatty-like substances can cross it. And these heads aren't to scale. They're actually very, very small. But things like fatty acids and cholesterol can go right through it because those are non-polar, uncharged molecules, so they can cross right through this, no problem. But what about if you need calcium to come across, or sodium to come across, or glucose to come across? This is a barrier. All those are either charged or hydrophilic. You can't get across it. So your body has developed a number of different ways of getting things across. So that membrane structure of the bilayer lipid membrane, this inner core right here, this lipid bilayer in here, it repels hydrophilic, lipophobic, polar, or charged molecules. At the same time, it will admit, this center will let pass hydrophobic molecules, lipophilic molecules, nonpolar, uncharged molecules. the physical chemistry of all these go together? The physical chemistry here of all these go together. Are they exactly synonymous with one another? No. Are they physically related to one another? Absolutely. So, sometimes you need to get molecules across the membrane, sometimes you need to get information across the membrane. So how do you communicate across this membrane if there's this barrier here? What if there's a molecule that needs to get across and it can't? Well, there's membrane transport proteins. So, take a look, and there's membrane translocation channel proteins. So, here's first is a membrane transport protein. So here is a very specific protein that has a binding site for a specific ligand. And a ligand, we're going to, well, ligand is all sorts of different things. It's just something that will bind this specific receptor site on this transport protein. So it binds there, and that transport protein, what does it do? It transports it across that barrier. That's what transport proteins do. Now sometimes, so these are all strategies the body's figured out how to get across this membrane. Sometimes you get molecules across, especially ions, by what are called membrane translocation channel proteins. So here's a protein, it has a channel that can either be open or closed. And something signals that channel to open. And these channels tend to be very specific. Some are just for sodium, some are just for potassium, some are just for calcium, some are just for glutamate. They tend to be very specific in what they let through. Let's say that we have a chemical concentration gradient, a lot of this green guy out here, very little in here. Now we have an open water-filled channel that it doesn't have to get past these fatty acid tails, it'll just go through. Those are membrane translocation channels. So transport proteins, port, P-O-R-T, means to carry. Trans means across. So transport proteins carry a protein across. Translocation just allow, they're an open channel, an open pore that allows something to flow down its chemical concentration gradient. Now these become very important because there's a lot of drugs that impact translocation channels. There's a lot of drugs that impact transport channels. So you don't just talk about these just to drive you crazy. There's a lot of pharmacology you're going to learn from these things later on. Sometimes you don't need to get a molecule across, you just need to get information across. So let's say this is a hormone, a growth factor, a cytokine. It can bind its receptor in the membrane, which in turn might activate, for example, a G protein, which will then activate a catalytic enzyme. You don't need to worry about writing all these out because you're going to get it later. And what does it do? It generates a second messenger. That was the first messenger. This is the second messenger. This will now tell the interior of the cell what to do and when to do it. That was the original extracellular message, and it set off a membrane signal transduction process to generate an intracellular messaging molecule to tell the cell what to do and when to do it and how much. So there's a lot of strategies the body's evolved to selectively deal with that barrier. You want that barrier because you want cells to be intact and protected. But you also need to be able to get select molecules in and out, or information in and out. So this is what we're going to be talking about for really more than the next hour. Not today, obviously, but I do want you to remember that the phospholipid heads, they're charged. Water happens to be a charged molecule. That's why membranes work so well, because yeah, these fatty acid tails, they hang out together. These charged heads, they want to hang out with water. And you've probably heard, you can take a solution of phospholipids, put them in water, shake them up, and you'll spontaneously form membranes. Why? They're just hanging out the way they want to in the lowest energy state, okay? So, this is something you really want to remember for a long time, and I've already mentioned it. Charged molecules like to hang out with charged molecules. Neutral molecules like to hang out with neutral molecules. So, The anti-corollaries of those are charged molecules are repelled by neutral molecules. Neutral molecules are repelled by charged molecules. It's like, oh boy, there's just a whole lot of jargon. You know, get this into your bones and you just really understand this. Now, this is where I really knew I had to get to, at least here today. But here are terms in physical interrelationships. Charge versus neutral. Neutral means uncharged. So charged molecules are polar. Neutral molecules are nonpolar. Polar just means there's a separation of charge. One part of the molecule might be neutral, the other part. There's just an electrical charge in it. Charged polar molecules are hydrophilic. They love, philic means love, right? Hydro means water. So charged particles like hanging out with water. Neutral molecules. Neutral molecules are things like fatty acids and cholesterol, things like that. They are hydrophobic. They fear water. They don't want to hang out with water. Things that are hydrophilic are lipophobic. Things that are hydrophobic are lipophilic. So here's some things that can freely cross a membrane without any help. Okay, some things that can cross, some things that can't. So